Hello, everybody. Welcome back to the Intellectuary channel. Today, we are going to discuss enzymatic functions and applications. And this concept is going to be found within the AP Biology course and exam description. And so, this is my first video within my new AP Biology course on my YouTube channel. So, today, at the end of this lesson, you'll learn to define what an enzyme is, how it can be utilized, and for what specific purposes it can be utilized for. You'll also be able to explain how a substrate binds to an enzyme and describe enzyme denaturation and the conditions of other enzymatic processes. You will also be able to recognize enzyme function in different environments of pH and temperature. And then lastly, you'll be able to evaluate different real world applications of enzymatic processes. So let's get started. So I want to start with this question of what are enzymes, right? So enzymes essentially are proteins, are types of proteins that speed up chemical reactions. So within enzymes, there's two things to remember. There's the active site and the substrate. Now, within the active site, all enzymes usually have an active site and a substrate that binds to the active site. So within the substrate is basically what binds to the active site of the enzyme. And when it binds to that active site, it often changes the shape and therefore the function of the enzyme. So here's just a basic image of this process of the substrate enzyme when it binds to the active site of the enzyme. So the green kind of thing is the substrate. The gaps on top of the circular object surface is the active site, and the circular object, I guess you can say, is the enzyme. So you can see how first there's a green substrate that is trying to attach to the active site or bind to it. And then the enzyme slightly changes shapes as the substrate binds to the active site. And where it binds is also called the enzyme substrate complex. And then the enzyme, within the enzyme, the substrate has already binded to the active site. And so now products are released from this reaction, basically. So products leave the active site of the enzyme in the last step of this enzymatic process. So here, enzymes can often be classified into different types of protein structures because they are a type of protein, and seen below. So we can see here a couple of them: quaternary and tertiary. So within the quaternary structure, so enzymes or proteins that are made up of multiple or several polypeptide chains are classified as having a quaternary structure. And here, basically, polypeptide means a long chain of amino acids linked or bonded together with peptide bonds. So, in the image to the right of the quaternary structure description, we can see here this image of this quaternary structure where this depicts the quaternary structure of an enzyme with several polypeptide chains connecting everything together. So, you can see this big structure over here. Moving on to the tertiary structure of a protein, enzymes which are in their final 3D shape or structure, and those who have a final folded shape, which is caused by the fold in the entire polypeptide chain, are usually classified as having a tertiary structure. So on the image to the top, you can see that image to the left depicts the tertiary structure of tryptase, which is an enzyme with the final 3D structure altogether and an entire polypeptide chain folded together, right? You need to have the entire polypeptide chain folded together, as well as a final 3D structure for you to call any protein or enzyme to have a tertiary structure when compared to quaternary or other protein types of structures. Moving on to the environment on enzyme function and efficiency. So here we have a couple of kind of scientific graphs where the y-axis in the first graph to the left, we see the y-axis represents the rate of reaction and the x-axis represents the pH level. So here we have three enzymes that were tested: pepsin, salivary amylase, and pancreatic lipase. And these two enzymes are used in different parts of the body and different other applications elsewhere. But we can see that there is a gray dotted line that goes between the middle of each enzyme's kind of graph, if you can see. And so that gray line represents where the optimum pH of the pepsin or some salivary amylase or the pancreatic lipase, that gray line basically represents the optimum pH, right? So wherever that gray line meets the x-axis, which is the pH level, that is the optimum pH level of the enzyme depicted. So we can see for pepsin where the optimum pH is. We can see for other enzymes here as well. And we can see that the rate of reaction is the greatest at when the enzyme is at its optimum pH level. So we can see here for the enzyme pepsin, it starts slowly and goes up really fast, the rate of reaction, and then it stops at the highest rate of reaction when it is at its optimum pH level. And then it goes down, goes down, goes down. So the rate of reaction goes down, 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 and then and farther away, basically, when it's farther away from its optimal pH. And we can see that trend continuous within both salivary amylase and pancreatic lipase enzymes as well. So that's an important trend to notice here because this is something that is obviously found in the CED or course and exam description within the AP biology course. And so, yeah, this is an important trend to remember because in FRQ questions, they might give you a graph specifically on enzymes and say what factors cause this or what is the optimal pH of this enzyme. And you can go to the graph and look at these factors that are affecting the rate of the reaction and say different things about it. So this is really important to notice. To the right, we have a big graph or a big graph depicting four graphs, we can say, where we can see here the first graph to the top left represents, again, the optimal pH levels on the x-axis, the rate of reaction in the y-axis, and the second graph we see temperature because not only pH levels affect enzyme function and structure and efficiency, basically rate of reactions, but temperatures also have an important role in affecting enzymes and their functions in different environments. So here we see that um, after the enzyme is going farther away from its optimal temperature, the enzyme starts to denature. And we'll learn more about enzyme denaturation in a further slide within the slideshow. So keep in mind. And then to the graph at the bottom left, we see that the rate of reaction, there's a proportional increase seen to the amount of substrate. So whatever amount of substrate there is, that much, I guess, proportional rate of reaction is going to be there. And then you see it slowly goes up, and then it just it's saturated because it's constant because the active site is saturated within the enzyme. And we can see the x-axis now doesn't represent pH or temperature; it represents the substrate. So that's how the rate of reaction has a direct correlation or relationship with the amount of substrate there is within an enzymatic reaction. To the graph at the bottom right, we see that the x-axis is now enzyme and the y-axis is rate of reaction. So we see that there's a proportional increase in the beginning, right? It slowly starts increasing to the amount of enzymes there are. And then because there is limited available substrate, the rate of reaction is going to slow down and just become constant or not increase or decrease because not much substrate is going to be available, right, to attach to several of the enzymes that are there. And if there's not much substrate to attach to the enzymes, then the enzymes can't really produce anything based on these processes, right? So that's also an important fact or takeaway to take from these graphs here. Now, here are some key points that describe the several graphs shown in the last slide, conclusions and descriptions, basically. So enzymes work better in environments with their optimal pH levels. And enzymatic function is in fact determined by the environment the enzyme is in. So excessive acidic pH levels can cause enzyme denaturation. And in colder temperature environments, enzyme activity is often slowed down. And in hotter temperature environments, enzyme activity is often sped up to a point where it depends on either the amount of substrate there is present within the enzyme, I mean, to attach to the enzyme's active site, or it depends on the amount of enzymes there are to speed up the chemical reaction. And if it's an extremely hot temperature, then that can also cause enzyme denaturation because it's excessive and it might be a lot further away from an enzyme's optimal temperature. So let's talk about enzyme denaturation. So enzyme denaturation is basically when an enzyme loses its functional 3D shape, making it less functional, often either losing its function or not being able to function properly because there is an alteration or some sort of change to the shape of the enzyme. So these are some factors that affect enzyme denaturation or make denaturation happen. So one factor is extreme temperatures, right? Temperatures that break weaker bonds within the 3D shape and structure of an enzyme can cause it to denature. Now, low temperatures in an environment most of the time do not denature the enzyme, but this is a usual trend, and we can't really call this as completely definite in all situations. And another factor is extreme pH levels. So within pH levels, if an enzyme is in an environment with a pH level that has a very high difference in relation to the enzyme's optimal pH level, then that can cause the enzyme to denature. So this can also mean, for example, extreme pH levels, such as extreme acidic or basic pH levels, can cause an enzyme to denature. And this all depends on how far the pH levels that an enzyme is in are from the enzyme's optimal pH level. So now that we've talked about enzymatic function, denaturation, and stuff like that, let's move on to competitive versus non-competitive inhibitors. So to the right, there's a Venn diagram that describes the uh, kind of conclusions that can be made about both competitive and non-competitive inhibitors and the similarities and differences between them. So within competitive inhibitors, right, their job is basically to bind to the enzyme's active site directly. And so when they do that, they block the active site from the substrate. So the substrate cannot bond to the active site of the enzyme. So this slows down enzyme reactions because it's not the substrate that's getting attached or bonded to the active site of the enzyme, it's the competitive inhibitor. So that essentially slows down enzymatic reactions. Now, when talking about non competitive inhibitors, its job is basically to attach to an allosteric site, which is basically somewhere else on the enzyme, different from the active site. And so when this happens, when this non-competitive inhibitor attaches to this allosteric site, which is basically somewhere different from the active site of the enzyme, the structure of the enzyme is impaired and it cannot function as well as it used to if the substrate or a competitive inhibitor were to attach to its active site. So here is a real world application of enzymes. So enzymes in the human body. So enzymes are crucial in a lot of ways, particularly in our human body. Enzymes such as amylase break down carbs and or starches, and lipase breaks down fats and lipids as well. And because of these enzymes, our body is able to properly digest our food, and that too at healthy rates of digestion. Here, keep in mind, rates of digestion have several direct correlations with enzymatic rates or rate of reaction, where the reaction here is digestion, and the enzymes participating in this process of digestion are lipase, amylase, and more. So, analyzing and being able to apply biological and chemical concepts that we've learned within the course to actual real world applications is really important because on an FRQ question, they might ask, what is a real world application of this concept that you've learned? And you'll need to be able to apply that concept to a real world application. And so within enzymes, proteins, and enzyme structures and function, you can say that this is a real world application that where enzymes are properly used in the human body, right? Because they are, and there are a lot of enzymes that help do so. So here are some review notes on this lesson of enzymes today. Where again, an enzyme is a type of protein that aids as a catalyst to speed up chemical reactions. And this is really important fact as well. They help in decreasing the activation energy needed to start a chemical reaction, because there needs to be at least some energy to start a chemical reaction. And so, by utilizing enzymes to start chemical reactions, it'll decrease the activation energy needed to start a reaction itself. Within environment on enzymatic function, we've discussed that environments in enzymes in, specifically extreme basic pH levels or extreme alkalinity, I guess, extreme acidic pH levels, extreme hot temperatures can all lead to the denaturation of an enzyme. So this makes the enzyme lose its function and 3D shape. We've also discussed enzyme structure. So enzymes are types of proteins that can be categorized into different protein structures. Usually the most common they're categorized into include tertiary and quaternary protein structures. And then we've also discussed factors affecting enzymes and their function. So non-competitive inhibitors and competitive inhibitors, the place of the active site of the enzyme, the environment the enzyme is in, right? pH levels of the environment the enzyme is in, the temperatures of the environment the enzyme is in, all are contributing factors that affect the enzyme structure and function. So everything we've learned within this presentation and within this video really is helpful to your understanding of enzymes and basically the AP biology course where it discusses enzymes and proteins. And so if you take good notes on this, you can probably understand a lot of MCQ questions or at least a few FRQ questions which discuss enzymes. Again, thank you, and don't forget to like, share, and subscribe to the introductory video so you don't miss any updates when I post new videos within my new AP Biology course, which I'm going to be doing on my YouTube channel. And it's based on the course and exam description, so it's going to be directly correlated with the AP Biology curriculum as well in the AP exam. So, thank you, and see you later.